Okay, hello, good morning, everyone. Thank you for joining. Today, our webinar is presented by Dr. Shuibing Chen from Cornell University in New York. The webinar is hosted by Greenstone Biosciences. And just before the webinar begins, just a few slides for introduction to Greenstone Biosciences. We are a biotechnology startup company in Palo Alto, California. We're a drug discovery, digital biology, and contract research organization. We do have a couple of compounds that are in clinical development, moving towards phase 1 clinical trials. One is for an indication of fibrosis, and another is an anti-inflammatory indication compound. At Greenstone Biosciences, we utilize patient-specific induced pluripotent stem cells that are isolated from patient blood samples. We've procured and licensing from Stanford University a very large and diverse iPSC biobank made up of over 2,000 patient iPS cells. And we use these cells for patient-specific medication testing and patient-specific toxicity testing in the preclinical setting. As I mentioned, we have the world's largest iPSC biobank that's been procured predominantly through Stanford University. We have over 2,000 patients in this hiPSC biobank representing many diseases, including cardiovascular diseases, musculoskeletal, oncology patients, and many others that can be used for multi-omics analysis, taking diverse populations into account. Our products range from cells, iPS-derived cardiomyocytes and other cell types, as well as three-dimensional organoids that can be used for high-throughput drug screening, testing a wide range of therapy modalities from small molecules to biologics, genetic therapies, and even radiation therapies can be tested in these in vitro human systems. We also operate as a contract research organization. We have cutting-edge, high-technology laboratories in Palo Alto with complete tissue culture suites and molecular biology facilities and equipment. If you're interested in working or partnering with us, these are our contact details. And without further ado, now I'd like to welcome today's webinar presenter featuring Dr. Shuibing Chen, leader in stem cell biology and regenerative medicine. Her pioneering work has advanced the field of pluripotent stem cell-based disease models and strategies, and also has promoted translational science, translating benchtop work to clinical translation. So without further ado, I'd like to welcome Dr. Shuibing Chen for her webinar. Thank you, Shuibing.
Can you hear me well?
Yes.
Okay, thank you for invitation. It's really my great honor to have the opportunity to present at the Greenstone Bioscience webinar. This is my disclosure. As many people in the audience, I'm trained as a stem cell biologist. In the meanwhile, I also have some chemistry background. So we are interested in two major topics. One is how to build the functional human organ models. In this, more recently, it's called we use organoid models. And the second question is if we can build it, how to apply them for disease modeling and drug screening. So just very brief introduction about human pluripotent stem cells. There are two types of human pluripotent stem cell, human embryonic stem cell and iPS cells. And they the reason it's called pluripotent stem cell because they have the ability to differentiate into in theory any type of the cells in the human body. So we at the beginning we were in the last 2 decades we spent a lot of time trying to understand the differentiation protocol strategies how to convert these single pluripotent cells into very complex tissue organs. And after 2 decades work, in addition to some protocol we develop in-house, we also adapt many protocols that developed by the field. So currently we have a platform, we can make more than 14 or 15 different type of cells, organoid, from human pluripotent stem cell, including like For mesoderm, like macrophage, peacemaker cells in the heart, and cardiomyocyte, which Dr. Zhou is really the leader of the field, and endothelial cells. And for definitive endoderm derivative, we make every alveolar for COVID study, small intestine colon cells, and pancreatic endocrine cells for type 1, type 2 diabetes, liver organoid. And recently, we also expand to ectoderm lineage, which we work on the cortical neuron, dopamine, and more recently, hypothalamus neuron as well as the organoid setting. So after we build all these tissue organoids, the next question is how to make it more pathological, physiologically relevant. So we spend a lot of time to build this kind of vascularized immuno-organoid model. There are usually two approaches people try to incorporate the vascular system and immune system in the organoid. One is like what Jim Wells is doing, more like co-development approach. Of course, it has been mimicked the developmental process very well. Because for our case, we are more interested in the drug discovery part, so we want to very precisely control the endothelial cell and macrophage components. So in this scenario, we use a reassembling approach. We make endothelial cells using ETV to overexpression, differentiate the macrophage, and we also make endocrine cells. Here's just one example how we do this assembled system. We have endocrine cells, endothelial cell, macrophage, and we reaggregate them together. After 48 hours, they will form this we call vascularized macrophage islet organoid, VMI organoid. And if you compare with primary human islets, they show very similar size and very similar size. And if we use confocal imaging to take a closer look, as you can see, they have this insulin green color label, the insulin positive beta cells, the red color label CD68 positive macrophage, and the blue color label the CD31 positive endothelial cells. So after that, we want to know whether the cells are just simply assembling together or they actually educate each other to make them better. So we monitor the cellular functions. And first, we start from beta cells. The major function of beta cells is to secrete insulin upon glucose stimulation. So here you show the figure we call dynamic GWAS. We culture the cells, organoid, in either low glucose to high glucose and back to low glucose and KCL. And we collect sample every other minute and run the ELISA to monitor the insulin secretion. And if you check the orange line, we can see the cell can respond to high glucose stimulation, go back to normal, and respond to KCL. If you put the same amount of endocrine cells in the VMI culture, in the red line, you can see the cell responds much better than the cells itself. And in the meanwhile, they also respond better to KCL stimulation. So this is the beta cells, and we also run a single nuclear multi-omics analysis to compare the co-culture cells and VMI organoid. If we separate the RNA and ataxic modality, the cluster 0 is the beta cells. And if we check the beta cell markers we find in the VMI organoid, the non-beta cell markers significantly go down, suggesting they are more biased to real beta cells. And the beta cell markers, such as insulin, like PEC6 or SLC2A1, they significantly go up, suggesting they are becoming more mature. So these are the beta cells. How about the other type of cells? We also check the endothelial cell. Endothelial cell, they have a very unique structure in the endothelial cell in the islets. They have a very unique structure called fenestration window. So that means the big hole on the membrane of the endothelial cells. So if the macromolecule like insulin, they can go through this fenestration window to enter the bloodstream. By running the electron microscopy, you can see this fenestration window on the endothelial cell human islets. In the human, at the right, you see the human ESC derived endothelial cells. The surface is very smooth. You see the in the VMI endothelial cell of the VMI organoid, we start to see this fenestration window formation, suggesting it's not only endothelial cell macrophage educate beta cells, the beta cell can also educate endothelial cell to make it more like endothelial cell in the tissue-specific endothelial cell. And lastly, we check the macrophage. The major function of macrophage is phagocytosis. So in this case, we run a live imaging analysis. The insulin, we use a insulin GLP cell line. So the beta cell was labeled with green color. And the macrophage was labeled with RFP, which is a red color. And we gave this organoid some Coxsackie virus B before, which is a virus isolated from pancreas or type 1 diabetes patient. So it can strongly infect the beta cells. And after the infection, the beta cell die, and now you can see the macrophage got activated. There is a enlarged macrophage, and they got bigger, and if you check it closer, you can even see some GFP residue still remaining in the macrophage. So using this assembly approach, we have built different type of organoid like vascular immunoorganoid, islet organoid like airway alveolar organoid, and a cardioid, and happy to talk more about that part. And after we make more different type of organoid models, right? The second question is how to apply them to disease modeling and drug screening. So 3 years ago, I got very fortunate to get support from Cornell. We built this center called Center for Genomic Health. And the concept is very similar to Greenstone. So the CGH is centralized using human PSC derived organoid model. We call it Organoid Village program. So if we call village, we have organoid human PSC and village from both male and females. Different genetic background, different disease background. More recently, people want us to make some organoid iPSC from like spinny mice. So we will expand it to animal platform too. And then after we make this organoid, we have 4 branches to try to set up collaboration applications. And the first branches is our single cell, our bioinformatics pipeline. And the reason for that is, when we talk with our clinician or surgeons, say, oh, we have very nice organoid model. Do you have any hypothesis to test? And they usually tell us, oh, we don't really hypothesis, but we do have a lot of samples in the biobank. So you can clearly see there's a big gap between the resource and knowledge. So what we do is to put together this bioinformatics pipeline, including single-cell multiomics, long-read multiomics, and spatial multiomics, including the long-reads. So by doing that, we scan a lot of samples, generate the hypothesis, and further validate it in organoid model. And I will just show you one small story that we did during the COVID time. And second pipeline is more like working with epidemiologists. So after we finished the first human genome sequencing project 30 years ago, now a lot of effort has been done for the GWAS, genome-wide association study. We have a long list for many diseases. We have long list for genetic factors, for genetic locus, or even genetic variants. So the next question is, which one is important, right? Which one is causal? So we do need a platform to study that, and I'll show you another example on that case. And the third branch on the bottom left is more like, I was trained as a chemist, chemical biologist. So we actually also develop a lot of chemical biology tools to the stem cell community. Unfortunately, I don't have time to talk about it, but recently we report a natural amino acid probe system to help you to put down any protein bind to any site of the human genome locus. So if you're interested in that, we'll be happy to talk and see what we can help. And the last part is drug screening. I think that's something we're always interested in. And then more recently, I got very fortunate to co-lead several NIDDK funded AI models. We'll touch base a little bit. Okay, so the first story I will tell you is on the data analysis, on how do we work closely with our clinician surgeons to collect information, build the organoid model, and discover drugs. So this story really is a COVID story. This you might recall by 2020, when by March, very unfortunately, New York City became the COVID pandemic center. And at that time, we immediately assembled a very large team at New York City across different institutions like Cornell, Sinai, NYU, Memorial Sloan Kettering, and Columbia. And we are very fortunate to work with many experts in the field. Like we work closely with Luke Kennedy on the chemical screening. Todd Evans is pioneer in the cardio differentiation. Doug Nixon is an expert on the HIV or viral infection, and we work with Rob Sports very closely on liver as well as biobanking for samples. And Dr. Heidi Stuhlman is an expert in the placenta development, so we do study the pregnant tissue a little bit. Dr. Herron, I don't think he needed any introduction. He has been very, very generous to help us on set up the long organoid model from the very beginning of the project. And Dr. Herron Westin is an expert in computational biology, so we actually test some computation, small molecule, they do the computation from the computational design. Lawrence Studer, I think another person doesn't need any introduction to this community. We work very closely on the neuronal ectoderm part of the project. And because we don't have any BSL-3 facility at Cornell, so we work with Dr. Ben Hanover at Sinai, who recently moved to NYU, and Dr. David Ho at Columbia to do all this virus work. So the story is, if we can make different type of cells, organoid in vitro, if there is a new emerging virus, what we can do. And we ask 4 questions, 3 questions basically. One is permissiveness. So which type of cell organoid can be infected by this new virus or bacteria? And the second question is, if they got infected, how do they respond? And the third question is, how immunocomponents contribute to that. As we all know that direct infection is only very small part of the whole story. And after direct infection, the immunocell come in and to further damage tissues. And for any of this work, we want to adapt it to drug screening platform to identify new drugs. So for the permissiveness, I will say a little bit surprisingly, for many type of cells we made in the Petri dish, they can be infected by SARS-CoV-2, but the infection rate highly varies. For example, lung, we see very high infection, and Doppler neuron, we see very, very low infection compared to other tissues. So, but we also collect a lot of biospecimens to validate the detection of at least viral protein in the tissues we found the changes in the in vitro. So after that, since we have this different organoid model, so they give us a very good opportunity to study the host response. If we just use viral cells to study virus, and the only thing is they got infected, they die, right? But when we take different organoids, run the RNA-seq or single-cell RNA-seq to study the host response, we do see very different response. For example, when we infect the lung airway organoid, we see a very high inflammation signal come up. When the endocrine organoid got infected, we found the beta cell, they lose insulin expression and gain the alpha cell marker like a glucagon. So the cell goes through trans differentiation. And for the pacemaker, I say nodal cells in the heart, this is the only cell type we see a very strong ferroptosis come up, and we still don't understand why. And 2 years ago, we published a last paper on the direct infection is with Dr. Lawrence Studer, is we find that Doppler neuron can be infected very, very low efficiency. However, when they got infected, they go through senescence. And it's actually a pretty interesting story because when we submit the cells to our BSL-3, and the standard protocol is they will take the cell out, do all this BSL-3 clearance, and then we do the data analysis. And for that particular case, the postdoc forgot to bring the sample out, fix the samples after 48 hours. And after 5 days, they ask whether you still want the samples, and we say yes. And indeed, we find they don't die, they actually go through the senescence process. So, and when the Doppler neuron got senescent, they can actually spread the signal, senescent signal to the brain, which may cause some brain damage. And then after this direct infection, and the next question we want to ask is whether we can build some immunomediate host damage model. So this is a large collective project with, as you can see, we have 5 postdocs co-lead the project. Liu and Yulin working on the whole drug screening part, Jiajun lead the effort of cardio differentiation as together with Fabric, and Ben is take care of all various work. So we start this idea is at 2020 unfortunately we have a lot of COVID patients in our ICU. When I talk with my colleagues at ICU, they told me that they see a lot of patients, they develop arrhythmia. And we immediately collect some samples from the healthy and COVID heart. At that time, we don't have resource to do single cell, so it's bulk RNA-seq. And if we check the cytokine chemical expression, we find that the CCL2 significantly upregulated in the COVID samples. And CCL2 is a very famous cytokine to trigger the migration of monocyte. So next we do CD163 staining for the macrophage, and indeed in the COVID samples we see a significant enrichment of CD163 positive macrophage. So next we set up this co-culture system from human PSC derived cardiomyocyte and the macrophage, and then we either only expose the cardiomyocyte to SARS-CoV-2 or we expose the cardiomyocyte plus macrophage to SARS-CoV-2. And the macrophage is from a MYG6 cherry line, and yeah, this is the line we made in the lab. If you're interested, happy to share. And if we monitor the ROI signal, on the upper panel, there's no macrophage, we see very low ROI signal. And the bottom panel, with the macrophage, we see an increase of the ROI signal. So we decide to run the high throughput screening. This is the most complicated screening of my whole career. We start from human ESL and macrophage co-culture, and then we treat them with FDA-approved drugs, and then send it to BSL-3 facility to run the infection. And 24 hours later, we fix the samples. After the clearance, we do the RS staining. So this is the primary screening data. If you run a lot of protein-based screening, maybe the Z-score is suboptimal, but because of the complexity of the screening platform, we got it's reasonable. And luckily enough, we find 2 hit compounds. One is called ranitidine, the other one is called tafasitamab, which actually can significantly decrease the ROI signal, the IC50 around less than 1 micromolar. And here's some primary screening data. As you can see in the ranitidine or tafasitamab-seated condition, we rarely see the red color ROI signal. And to summarize this part of the story, we find that some type of cells in the heart can be infected by SARS-CoV-2, and then they secrete CCL2 and then trigger the monocyte to migrate in. And they will differentiate into macrophage and secrete TNF-alpha and IL-6, which damage the cardiomyocyte. And when we run the drug screening, we find 2 compounds. One is tafasitamab, is a JAK-STAT inhibitor. The other one is ranitidine, which is a ROS inhibitor. Both can protect cardiomyocyte from macrophage-mediated damage. And when the paper was published, coincidentally, FDA also issued emergency use of JAK inhibitor on COVID patient, and later the phase 2 and 3 clinical trial also saw very promising results. Hopefully, we don't need to use this drug very soon. For this purpose very soon. So I will move out from the first example about how this analysis of patient samples can give us some idea about hypothesis, generate some hypothesis and validate the organoid model. Now I move to the second branch of what we are doing at CGH is how do we use these organoid models to study the human genetic signal. So we start this part, I will move back to beta cells. Starting, we have been interested in this question for a pretty long time. Back to 2014, at that time we have 2 very talented postdocs, Mingguo and Huizheng. So they want to ask whether the human ESL derived system is good enough to study GWAS identified hits. You might recall at that time, many people are still working on monogenic disease. And They choose 3 genes, CDKL1, KCNG11, and KCQ1. And they choose these 3 genes because they are the first wave genes that identify the first wave of the GWAS study and validated in the follow-up studies. And it's also highly expressed in beta cells. So they first use a CRISPR technology to knock out the genes, create the isogenic line, and differentiate into beta-like cells. And they monitor their survival function in vitro. On the left, you see the human insulin secretion data. When we work on the isogenic system, we always need at least 2 clones to consider clone-to-clone variations. And for wild-type cells, it's a low glucose condition, we can see some C-peptide secretion, and the high glucose condition, we see much higher C-peptide secretion. And for CDKL1, KCNQ1, KCNG11 knockout lines, they completely lost the response to glucose stimulation. And in the second part, we also monitor cell survival. In this case, we culture them in high glucose or hypometry condition, which is type 2, mimic the glucose lipotoxicity in type 2 diabetes patients. I know it's a very busy slide. I will directly go to the quantification part. When we compare wild type and CDKL1 knockout cells, in the control condition, there is no difference in between wild type and knockout cells. However, if we challenge them with high glucose or hypometry condition, the cell cannot handle the stress very well, they die more. So this is second phenotype we show. And finally, they transplant this human ESC derived beta into the kidney caps of the immunodeficient mice. And 4 weeks later, for the mice transplanted with wild-type cells, we can see very nice low insulin secretion and low fasting condition and very good response after glucose stimulation, consistent with our in vitro result. The CDKL1, KCNQ1, and KCNG11 knockout lines, they don't respond very well. And after we set up this platform back to 2017, I met Dr. Francis Collin in the conference. Again, I don't see Dr. Collin in any introduction either. So he lead the human genome sequencing project. And after that, his own research has been really focused on the type 2 diabetes. So we are interested in the same question, right? After GWAS identify all these hits, they are all correlation analysis, how to determine the causality. And indeed, the genomics team, they develop a lot of tools. However, we still feel that we need a wide lab system, very high throughput system. We can do functional validations. So we designed a 3-phase project, and later we actually add another project, which is a little bit unexpected, but the result is very promising. So the first phase, we focus on the effector genes. We do the gene knockout. And the second phase, we move to the silence of the regulatory region. And the third phase, we are still designing to understand how to do perturbation for these 8,000 SNPs. And the third, the most recently set up model is we call a GWAS in dish model. I will share with you some preliminary data. So we start from the isogenic system. And this project is led by very talented postdoc Dongxiang Xue from my lab and very talented staff scientist Narasu from Collins' lab. So T2D currently they have around 256 effector genes, and Narasu did very careful computational analysis, check with multi-omics data across different tissues because type 2 diabetes is a multi-organ disease. And he finally narrowed down to 20 genes. Is based on the computational prediction, is most relevant with beta cells. So what Dongxiang did is individually knock out all these genes and create isogenic lines and differentiate into beta cells, monitor their function, and also run RNA-seq, ATAC-seq to do some integrative analysis. So these are the 5-cell trees. And when we make this reporter line, we use a MEL1 insulin GLP reporter line. And in this case, we can directly monitor the percentage of GLP positive cells as the differentiation efficiency readout. And when we monitor the differentiation, we find like knockout of 6 genes, the cell lose the differentiation efficiency. And next, we monitor the insulin production. So we basically purify the insulin GLP positive cells, and then we run ELISA. And this case, more than 10 genes, if we knock them out, we see an impaired insulin production. And whatever the lines, we see a statistically significant difference across both clones. It was labeled as red color. And the third assay we run is beta cell survival. So in this case, at the beginning, we only monitor the cell in regular culture condition, and we don't really see any difference across wild type and knockout lines. However, when we challenge them with palmitate condition, they cannot handle the stress very well. Again, as you can see, there are around 15 lines failed in the beta cell survival assay. And lastly, we run the beta cell function, which include the glucose stimulate insulin secretion as well as the KCl stimulate insulin secretion. And we find most of the line they fail in the glucose stimulate insulin secretion, and around 6 of them are more relevant with KCl stimulate insulin secretion. So if we put this together, this is a summary of the cell tree analysis. And one gene stand out is called HM4 alpha. It's associated with all 5 trees. And another name of HM4 alpha is called MODY1. It's the first gene identified to be related with monogenic diabetes. So it completely makes sense. And some other genes that can associate with 1 or 2 or 3 or 4 different cell trees. If we just stop here, we have a system. You can see we put 20 knockout paper in one, right? So usually the scale of the study can give us some opportunity to see something we haven't seen before. So in this case, Dongxiang isolate this insulin GLP reporter, insulin GLP positive cells, and run a paired RE-seq and ATAC-seq. And they are still analyzing the data, but this is some preliminary analysis. So if we consider each knockout as one individual, and for this isogenic system, we have a functional readout, which is the insulin content. And we also have the gene expressions. So Narisu ran this linear correlation analysis to find the genes that are either positive or negative correlated with the insulin production. And in this case, they find 2 genes that are positive correlated with insulin production and 3 genes that are negative correlated with insulin production. And Dongxiang used CRISPR-I and CRISPR-A system to validate 4 out of 5. So now they are moving to the second phase of the project. So by doing the computational based prediction or AI facilitated prediction, right now there are around 876 regulatory regions, which may be more relevant with the beta cells. And at this scale, making the functional data, make the individual knockout line become impossible. And in this case, Dongxiang used a CRAB system. So we designed every 50 base pair, we design 1 sgRNA to silence the regulatory region. And we use a pooled CRISPR-I screen. And in this case, Dongxiang find use 2 assays. One is insulin production, one is cell survival. She find around 15 different sgRNA, either positive or negative correlated with insulin content, and another I think it's around 40 sgRNAs that are either positive or negative correlate with beta cell survival. And currently they are running the single nuclear multi-omics analysis to see if we silence these individual regions, what kind of transcriptome, epigenetic, and cellular changes happen to these cells. And hopefully we'll be able to report more data on this part. And phase 3 of the project, we're still doing the experimental design. As you can see, it's a very ambitious goal for 8,000 SNPs. And if you have any good system to solve this problem, I'm very happy to talk. Last part is an independent project, but it's moved forward much better than we expected. We call GWAS in a dish. So I think we already introduced the genome-wide associated study concept, right? So in the human population, there are a case, in this case, maybe type 2 diabetes patient, and then control population, then we rule in either SNP array or GWAS, and run the correlation between different genetic variants with the disease progress, disease stage. And then we identify the genetic variant locus that is important for this disease, like Manhattan plot. If we think about the GWAS studies, if we check this blue bar, 20 years ago, any GWAS study, if we have like 10,000 samples, that's already very good. However, if you check the paper published in last 2 or 3 years, we start to read the patient with like a million patient or even more than a million patient. Which is good news is we have a lot of samples. However, human studies are very, very expensive. So we are wondering why do we need so many patients to generate the significant results? Well, one thing to think is like type 2 diabetes and also many cardio disease. They are many factors can affect the disease progression, like the lifestyle, what kind of food the people eat, and what kind of drug they take. So this environmental factor really cause a lot of signals so that we have to have large population to identify the real genetic signal. So in this case, we have several years ago, we have a collaboration project with Illumina. We do iPSC banking and whole genome sequencing for 500 type 2 diabetes patients. And here we use around 100 or 96 of iPSC lines. And we differentiate them into beta-like cell in the 96 well format. And indeed, they do show some variation regarding their differentiation efficiency. But when we challenge them with palmitate, like lipotoxicity condition, we see pretty consistent results regarding their survival rate. As you can see, there are many lines, they are pretty robust, so they don't really respond to palmitate, so the survival rate is around 100%. And there are some lines, they are very fragile, so the survival rate is less than 5 to 10%. So now we do the correlation analysis. Leland Taylor, postdoc in Collins lab, who ran this the correlation analysis between the genetic signal to the response to lipotoxicity as a single readout. Because in this system, we tightly control the reprogramming approach, the cell culture, the cell will culture differentiate in the same condition and exposed to exactly same dose of palmitase. So the environmental factor has been minimized. In this case, we can maximize the signal of genetic factors. And we find two locus. These two locus, they are highly correlated with survival rate upon palmitate-treated condition. If you work on human-based GWAS, you might laugh because the p-value is only log 10 p-value is only 4 of 5 or 7, but because of the size, we only have 96 iPSC lines. So it's pretty impressive. We already can find some locus that is important. So next, we want to validate our finding. We want to convince people this is really important. So we focus on, we decide to follow up on the locus. One of these locus is correlated with a gene called SPRY2. And Shuibing Chen, a postdoc leading this effort, first make the knockout lines. We have two wild type 2 knockout isogenic clones. Luckily enough, knockout this gene at iPSC stage doesn't affect differentiation efficiency. So we have pretty nice pluripotent cells. And then next, we check their differentiation to insulin GLP positive cells. And again, when we knock down these lines, SPRY2, we don't really see any effect on the differentiation. However, when we challenge these cells with palmitase, it's very similar with our primary screening condition. Here we see in the wild type, we only detect like 6 to 7% early apoptotic cells, but in the SPRY2 knockout lines, we see a significant increase of the beta cell apoptotic rate. And we also transplant these cells into the immunodeficient mice. And if you check the xenograft in the wild type, we see very little cleaved case-base 3 positive cells. And in the xenograft transplanted with SPRY2, we see a significant increase of cleaved case-base 3 staining. So then we want to know whether this related with human population study. If you go back to human GWAS, indeed, SPRY2 is a hit from the type 2 diabetes GWAS study. It's just not the most dramatic case because the signal might be affected by some other studies. But in our tightly controlled GWAS in dish system, this stand out as the top hits to affect beta cells' response to lipotoxicity. So at the last maybe 10 to 15 minutes of my talk, I will switch the gear to another topic, which is the drug screening. And originally I was trained as a chemist, so I love chemical screening and I run a lot of chemical screening in my own lab. At CGH, we actually have a drug screening pipeline, start from primary screening to hit confirmation, to dose curve, in vitro, structural activity relationship analysis, in vivo mechanism, and some of them patent. And this is an open platform. We would love to talk if you are interested in running some collaborations. So the traditional chemical screen is that if you're interested in beta cell function, we run one screen. Okay. If you're interested in beta cell proliferation, we run another screen with Ki67. If you're interested in beta cell apoptosis, and then we stand for the clean phase 3. And we plate the cells in 3-fold or 96 well plate, treat with chemical libraries, and then do immunostaining or reporter-based assay. People have been running this type of phenotypic screen for more than 2 or 3 decades. It worked well. We identify hits. However, it's just very expensive. For each assay, we need to have one screen. So Gia, a very talented student from my lab, so she asked a question whether we can do something a little bit smarter. So what she designed is she called this chemi-perturb-seq platform. We have 2 phases of chemi-perturb-seq. At the phase 1, we barcode the cells, and phase 2, we barcode cDNA, which is more efficient. And let's talk about phase 1. So in this case, Gia barcode different beta cells and then put it in 96 well plate. Each well is 1 barcode, and then we give them small molecules. And by doing these combinations, then we run the single cell. After several days culture, we run single cell RNA-seq. And by reading the barcode, we know which chemical exposure the cell has been exposed to. And Gia actually, because it's a very heavy database, Gia worked with Ricky and Han from University of Michigan to make a website to make the data public available. And I will try to do a quick demo. Here. Okay, the website is called ChemiPerturbDB, and I apologize, I'm working from home today and the Wi-Fi signal is not very good. So instead of showing the live demo, I'll just show you some results, but just play with it and let us know whether you like it. So I think most of you use ChatGPT or Gemini or maybe the Cloudy every day. So it's a very user-friendly system. It's more like a ChatGPT format, and you can just tap whatever question you have. And here, if you tap, ask what is PDX1, and then we have a fine-tuning model on top of ChatGPT. So they will give you some specific information about this transcription factor and what's their relationship with diabetes. And then you can ask, say, show me the expression of PDX1. And this time they will do some real-time analysis, which usually take around 30 seconds. So they will pull out this violin plot for each dot correlated with 1 cell. So, and here you can, and x-axis you see different hormones. And here you can see different hormone treated condition, they can change the gene expression level. This is single gene. And you can also tap, show me the beta cell proliferation. This time they pull out the beta cell proliferation pathway, and then they do the gene site enrichment across each individual DEG list. And they can find the hormone that can either upregulated or downregulated proliferation pathway. We can also do individual pathway. For example, recently we are working on Hippo. So if you say show me Hippo pathway, now they will extract the gene site associated with Hippo pathway. Again, run the GSEA analysis. In this case, we find there is only 1 hormone, glucagon, which downregulates the Hippo pathway. So in addition to this predefined pathway, there is also my favorite function is it's called advanced enrichment analysis. So in this case, you can put a gene list as short as 5 genes, as long as around 200 genes. And then if you launch, it takes around 4 to 5 minutes. So I won't do a live demo. And they will annotate the hormones or the compound that either upregulate or downregulate the gene you are interested. So it looks like pretty cool system. And I need to show you it's really work. So if I go back to the presentation, in this case, GR use this platform to help address questions related with islet transplantation. So islet transplantation, we all talk a lot about human stem cell derived beta cells it's already on phase 3 clinical trial. And islet transplantation was approved back to 2013. So when the donor passed away, they very generously donate their pancreas, and there are around 48 hours window. We can isolate the islets and transport it and infuse to the portal one of the recipients. And it has been on clinical trial for more than 20 years. Although it's already FDA approved, there are still 2 major challenges. One is cell survival. So more than 40% of cells die in the first 2 weeks post-transplantation. And the other question is the transplantation site. We put it in the portal one because it's close to liver. Liver is a major organ used insulin. However, anything we put it in liver, if anything goes wrong, there's no way we can take it out. So GR developed a project. We focus on subcutaneous transplantation, which is much safer side. If anything goes wrong, we can just cut it and take it out. So she first ran a barcode screen and find a hormone combination called lipotrophin IGF-1 and PG2. And it's 48 hours pre-treatment. The human islets show much better survival when she transplanted into the female mice. As you can see at week 1 in the blue bar, in the lip-treated condition, we already see increased insulin secretion, and the increased insulin signal keep increasing until week 4 and even longer. However, when she transplanted this model to male mice, unfortunately, nothing worked. So she was very disappointed, and then she extracted the xenograft, transplanted into a lip-treated xenograft, transplanted into male and female mice, and run a single-cell RNA-seq. And the first signal she finds is the TNF pathway are enriched in the xenograft transplant into the male mice. So she put the TNF pathway in the chemi-perturb-seq. And in this case, the chemi-perturb-seq annotate 4 hormones that can downregulate TNF pathway. And she tests each of them, and serotonin was found to increase the cell survival. And she was not really fully satisfied by that, so she also want to focus on the genes that are upregulated in the xenograft transplant in the female mice. This time she chose the top 20 genes, and she put these 20 genes in the chemi-perturb-seq again. And this time the system only nominates 1 hormone, histamine, that can upregulate this gene site. And when she do the combination of serotonin and histamine, and she find indeed this, if we add these 2 hormones together, even 1 week post-transplantation, when we put it in male mice, we already can see significant upregulation of the insulin secretion. And you may ask, is male and female that different? After we did very, very extensive literature search, GR find a 20 years old paper. They actually monitor the serotonin and histamine level in both human peritoneal cells and blood cells. And based on the results, as you can see, both serotonin and histamine, they are at least 2 times or even 3 times higher in female and than male. So that's explain why we need to add additional hormone pre-treatment before we transplant into the male recipient. So I think I will that's basically the scientific story I want to share with you for now. And for the at CGH, we generate a lot of single-cell RNA-seq multi-omic spatial data, and we spend a lot of effort to put them public available. If you do a screenshot, feel free to visit our website and let us know if you have any suggestion, comments. And I've been very fortunate to co-lead several NIDDK funded data science programs like DKNet to build AI tools, pen base to make a knowledge base, and also more recently multimodality AI for type 1 diabetes for large data integration. And lastly, as you're all aware that last year there was a lot of policy changes, and FDA have this Moderation Act 2.0 and 3.0 to phase out from animal testing. And NIH also claimed to prioritize the human-based research. And this is not only in US, right? EU is also phasing out from animal studies. So in response to that, I'm working with ICR to launch a new consortium called Advanced Stem Cell-Based Model in Drug Discovery and Development. And it's a global consortium including industry, regulatory agency, and academia scientists. We want to help to launch the global regulatory research priorities and try to accelerate the process to integrate stem cell models into preclinical stage of drug development as well as a regulatory agency process. And we have to say thank you to Joe and Todd, who very generously to help us to serve as the co-chair of our working group 1, which focuses on the preclinical drug discovery. And if you're interested in this consortium, please scan the barcode. It will direct you to the ICR website. And we are still putting the working group together, and your volunteer is more than welcome. I think this is the most important slides. I think I mentioned most people's name during the talk, and I want to thank all their contribution, all our amazing collaborators and our funding agency, and I'm ready to take any questions. Yeah.
All right, thank you, Dr. Chen. That was a hello.
Yes, I can hear you, but it's a little bit echo.
There's the echo.
Yeah.
Hello. Is that
Still a little bit echo, but it's okay. Not too bad.
okay? Can you hear me now?
Yes, it's much better.
Thank you. That was a great presentation. Very, very interesting, very informative, very timely with the regulatory changes that are happening at the FDA and at the NIH. So there are a few questions for you to field. So one question is about the cell village or the organoid village pertaining to the size. Is there a technical limitation as to how many patients can be represented in a cell village or in an organoid village?
Yeah, that's a wonderful question. I don't think I mean, you guys iPSC bank maybe even our than our current iPSC village. As has always been a question people ask me is how many iPSC do we need to run this type of analysis? The quick answer is the more the better. But if we're handling a large set of organoid model, I think it's need to be moved from the completely manually system to maybe automation system. So that is the limiting factors. And if 96 well played is the maximum we can handle in the lab at current stage, and I would expect for the field larger scale need to be moved to more industry setting.
Okay. Next question is about the perturb database, the DB that you showed the very nice illustration and demonstration of. Question is how to directly access that. Is it through your website or another?
Yeah, you just go to the www.perturbdb, and it's an open access data. Right now, we don't have any limitation. And a good news for this community is, right, in addition to beta cells, we recently also generate data for cardiomyocyte as well as a cortical neuron. So we are still analyzing the data. We want to do some validation to make sure we are confident about the data. And by that time, we will also release on the same website to make it all public available. Yeah.
All right, thank you. There's a question about cardiomyocytes. So in the illustrations that you showed, most of the cardiomyocytes had 2 nuclei or were binucleated. Is that something that you typically see for iPS or embryonic stem cell cardiomyocytes? And how does that compare to the adult hearts?
That's a good question. That's actually a good catch. I did not pay too much attention to that part. But my understanding is if we culture them long in longer culture, we do see some fusion things happen during the iPSC culture. Assume it's more closer to the mature beta cell, mature cardiomyocyte, which are multinuclear. And we because the protocol is already published, so just feel free to test to see whether it's better than than the other protocol to promote the fusion. Yeah.
Mhm.
Mm.
And a follow-up to that is if a cell has two nuclei, or the two nuclei identical, or can each nucleus have a different gene expression profile or a different epigenetic profile?
That's a very good question. I was thinking what's the best way to address that problem. Even single nuclear multi-omic may not be good enough, right? So we we do need to label the cell first so that they have the same barcode. And then we do single nuclear multi-omic analysis. If they have different nuclear barcode, then we can check carefully whether they have different genome or not. My expectation, I don't think it will be identical. I don't think it's come from the same division, the cell division. It's more come from perfusion, the fusion process. If it's come from the fusion process, maybe they will carry different transcript pattern. But that's my guess. I guess to answer that question very detailedly, we do need a barcoding system to barcode individual cells, and then we run single nuclear multi-omic, and we identify two nuclear with the same barcode, and then do a direct comparison.
All right. So the next question is about islet transplantation. The question is, can you test for immune rejection or immune compatibility in vitro prior to transplantation?
Yes, so I don't have time to talk about that. Recently, we spent a lot of time to set up the T cell, beta cell co-culture system. So we have very good model to test allogenic rejection, which is relatively easy. If they are HLA not matched, they put T cell, they die. And we are also putting some model. It's more like a CAR T approach. We overexpress autoantibody, the CAR that recognize autoantibody for the beta cell in type 1 diabetes. And then we can do this allogenic system to study the rejection. We are working on that.
All right. Then that kind of leads into the last question, which is a question about autologous versus allogenic cell-based therapies. And looking to the future, do you think there'll be a day when it can be done in an autologous way to avoid immune rejection?
Wonderful question. I know people are trying different approach, right? For beta cell replacement therapy, a lot of effort put on encapsulation, but we haven't seen too much progress yet. And recently, there are also effort published human islets from New England Journal of Medicine. They use a knockout approach like hypoimmunocells, right? Knockout HLA, B2A, and just different immuno-associated genes. Based on the Alex data, one single patient, it looks very promising. And I know several companies are taking that approach. To me, I'm maybe a little bit conserved. I still prefer the HLA matching system. I think the presence of HLA can still try to protect the transplanted cells. Just make them completely blank to the immunocytes make me a little bit nervous. But we'll see. I think different approach people are testing in next several years. We'll see a lot of data come out, and the data will tell us the answer.
Mm-hmm. Right. Okay. So that's the last question. I want to thank you again, Dr. Chen, for your presentation and for all the hard work that you've done over the years, contributing to regenerative medicine and to the use of pluripotent stem cells in vitro to study clinical science and translational sciences. Thank you very much. And a reminder to the audience, there will be a recording of the webinar today available on our Greenstone bio.com website for you to visit after the meeting. Thank you very much again, Dr. Chen, and have a wonderful day.
Yeah, thank you, everyone. Thank you. Have a nice weekend.
All right. Bye-bye.
Bye.